Today, I'm going to discuss with you people the second chapter of 11th standard biological classification. We have already started with the lecture. Now, this is my second lecture based on biological classification. Initially, I discussed with you people about this two system of classification, then comes the three system of classification, then the five system of classification, then the six system of classification. Now, let's discuss with the RHWhitakers five system of classification. In that, we are going to discuss today about Kingdom Monera. According to the five system of classification, the first kingdom is Monera, second one is Protista, third is Fungi, fourth one is Plantae, and the fifth one is Animalia. Next, now this lecture includes the characteristic features as well as the classification of Kingdom Monera. Let's start with it. They are the primitive organisms. Primitive organism means they are the first occurring organism on this earth. These organisms, they are prokaryotic in nature. They are present both in living as well as non-living environment. They can survive the harsh as well as the extreme climatic conditions like hot springs, acidic soils, etc. They can survive in the region where the other organisms cannot survive. They are the unicellular organisms. Membrane-bound nucleus is absent. They lack the membrane-bound nucleus, and the nucleus that is present in this prokaryotic monera is called as nucleon. That shows presence of DNA which exists as a double-stranded circular single chromosome. Along with this circular single chromosome, there is an extra chromosomal genetic element which is called as plasmid. So plasmid is present within the prokaryotic cell, that is Monera. Okay, next is the rigid cell wall is present. They lack the membrane bound organelles like mitochondria as well as some more organelles like endoplasmic reticulum, Golgi complex, etc. because these are the prokaryotic cells. That's the reason why they lack the membrane bound organelles. Habitat. Where is this Monera survives? They are found everywhere. They're omnipresent. It is like wherever the other organisms does not exist, there exists this Monera. For example, in hot springs, under ice, in deep ocean floor, in desert, and or inside the body of the plants as well as animals. Next, let's see the proper features of Monera. They are the unicellular organisms, or you can say they are the single cell organisms. They don't have a proper nucleus. Bacteria are included as a which as a component, or the bacteria, it constitute the major kingdom. Bacteria is an example, it's the sole member of the kingdom Monera. There are various forms of bacteria which is existing on this earth. Some bacteria, they are beneficial to us, some are harmful to us. Some monarians, they absorb nutrition from the environment and they are called as heterotroph. Some, they can photosynthesize on their own, and that means they can prepare their own food and they're called as autotrophs. Some monarians can move around while others stay in one place. Monarians, they're asexual, meaning they split into two, reproducing the identical offspring. Here you can see the structure of eukaryote and prokaryote. Here why I have taken this structure, because Monera is under prokaryotic organism. When you compare Monera with the eukaryotic organism, you can see that prokaryotic organism like Monera, they lack a well-defined nucleus, and that's the reason why it is called as nucleoid. They have certain locomotory organs such as flagella, which is absent in eukaryotic cells. Next, here is a slide showing you some of the examples of Monera, some organisms which are existing under the kingdom Monera. First one is Clostridium tetani. Next is Streptococcus pneumonia. Next, Staphylococcus aureus, Legionella pneumonia, Clostridium botulinum, Salmonella typhi, Vibrio cholerae, Treponema pallidum. All these are the organisms, the bacteria which exist under the kingdom Monera. Next, Monera is classified under two categories. One is eubacteria, which is also called as true bacteria. Second one is archaebacteria. Eubacteria is classified on the mode of nutrition under two categories. One is heterotrophic bacteria and the second one is autotrophic bacteria. Autotrophic bacteria is categorized under chemoautotrophs and photoautotrophs. So when you talk about the heterotrophic organism or heterotrophic bacteria, it means that they cannot synthesize their own food. They are dependent upon others for their food. That is what is heterotrophic bacteria. Then next one is autotrophic bacteria. Autotrophic bacteria means they can synthesize their own food. They can synthesize by using the process of chemo. They are the chemosynthetic organisms, or they can be photosynthesizing. That means chemoautotrophs, they're not going to depend upon the sunlight, but they're going to depend upon some chemical compound for their manufacturing of their food. That means they can synthesize their food from some inorganic substances. But at the same time, what is this photosynthetic bacteria? They can synthesize their food in the presence of light. That's the reason why you call it as photoautotrophs. The example of chemoautotrophs are sulfur bacteria, and the example of photoautotrophs are the cyanobacteria. Next is archaebacteria. Archaebacteria, depending upon their habitat, they are being classified under three categories. One is halophiles, second one is thermoacidophiles, and the third one is methanogens. Now, when you talk about archaebacteria, we can see these bacteria, they are mostly found in the extreme conditions. That's the reason why they are also called as extremophiles. That means they can survive in the extreme conditions. So they are called as extremophiles. Now, they are also found in high salinity areas where the saline condition is more. That's the reason why you call it as they can exist in salty area and you call it as halophiles. Then what is thermoacidophils? Thermoacidophils is they can resist the increase or the extreme temperature. That's the reason why they are thermoacidophils, as in the hot springs. Next is methanogens. Methanogens are those bacteria which help in the production of methane in the biogas plant. And that's the reason why you called it as methanogens. Now, let's talk about bacteria. The mode of nutrition in case of bacteria is autotrophic and heterotrophic. Autotrophic is divided into photoautotrophs as well as chemoautotrophs. Photoautotrophs, they uses light energy as a main source of food. Chemoautotrophs, they use the chemical reactions to derive their light energy. Next is heterotrophic. They depend on others for their food. They might be parasitic in nature or they might be saprophytic in nature. Parasitic are the ones which survive or which depend on other plants and animals, which live on or in other plants and animals. Those are parasitic in nature. Saprophytic are the ones which depends on the dead decaying organic matter. So this is nothing but what we call parasites and saprophytes. Next is depending upon their shape, the bacteria are classified under four categories. The first one, their bacteria, they are called as coccus. Coccus means the shape of the bacteria is spherical. Next, you call it as bacilli. Here, bacilli means rod-shaped bacteria. Next one is spirelia. Spirelia means they are spiral-shaped structures, and hence the name spirelia. You call it as spirelium as well. Next one is comma-shaped bacteria called as vibrio. You call this a comma-shaped bacteria, which you call as vibrio. You can even call it as kidney-shaped bacteria. Next is, if you see about cocci, it is coccus, which is single-celled. Then diplococci, they occur in pairs. Tetrad, which are group of 4 cocci. Next is sarcina, which are cube-like shape. Next one, streptococci, they are the chain-like morphology. Staphylococci, they are the grape-like clusters. So here you can see that the cocci, they are distinguished into in the form. If cocci occurs in single, it is coccus. If it occurs two in number, it is diplococci. If it is four in number, they are tetrad. And if it forms a cube-like structure, it is sarcina. And if it is streptococci, if a chain-like structure is formed, it is called as streptococci. If a grape-like cluster is formed, it is called as staphylococci. These are some more diagrams of the singly occurring cocci, monococcus. Then this is diplococcus. This is the chain-like structure called as streptococcus. This is tetracoccus in tetrads. Then you have staphylococcus, grape-like structure. Then you have sarcina, which is 3D structure, a cube-like structure. Next, we're going to talk about bacilli. Bacilli arrangement. In this case, the monobacillus, they remain single rod-shaped cell after dividing. That is, this is called as the single bacilli, monobacillus. Diplobacilli. This cell remains in pairs after dividing. So this is diplobacilli. Next is streptobacilli. These cells remain in chains after dividing. So this is streptobacilli. Next is palisades. Cells in chain are arranged side by side instead of end to end. They are partially attached. This is palisade. They are attached end to end. Next comes the coccobacilli. They are short with slight oval shape resembling both coccus as well as bacillus bacteria. That's the reason why they're called as coccus bacilli. Next comes again just a revision sort of for the types of bacteria along with their examples. If you talk about cocci, it occurs in diplococci form. Here the example is Streptococcus pneumonia. Then next comes Streptococci, Streptococcus pyogenes. Next is tetrad, and then comes the staphylococci. The example is Staphylococcus aureus. Next is the cube-like structure sarcina called as Sarcina ventriculi. When you talk about bacilli, it occurs in a chain-like structure called a chain of bacilli. The example is Bacillus anthracis. Then there are flagellated rods present on this bacilli. Example, Salmonella typhi. Then there are spore formation or spore former bacilli called as Clostridium botulinum. Then others which are comma shaped are the Vibrio cholerae. Then spiral structures, Helicobacter pylori. Spirocats, Tryponema pallidum. So these are the examples of certain bacteria with different shapes. Now, the next slide is the classification of the bacteria based upon the staining agent. The stain that is used over here is crystal violet. The bacteria is allowed to stain with crystal violet stain. All bacteria will be stained bluish or bluish purple. Then a mordant is used which is called as 0.5% iodine solution. The stain will be fixed by this mordant. Later on, destaining with alcohol or acetone is done. There are some species, bacterial species, in which the stain does not come out. At the same time, after destaining with alcohol or acetone, there are some bacterial species in which the stain will easily come out. The stain in which the species in which the stain does not easily come out, they are the gram-positive bacteria. The bacterial species in which the stain comes out very easily, these are the gram-negative bacteria. Next slide is showing you the classification of gram-positive and gram-negative bacteria. The gram reaction, in case of gram-positive, they retain the crystal violet dye and stain dark violet or purple color. In gram-negative bacteria, they can be decolorized to accept counter strains like safranine and they stain red. Next is peptidoglycan layer. In case of gram-positive bacteria, is a thick layered and multi-layered structure. In gram-negative bacteria, the peptidoglycan layer is thin and single layered. Teichoic acid in gram-positive, there are many in number. Teichoic acid in gram-negative, they are absent. Periplasmic space is absent in gram-positive bacteria. Periplasmic space is present in gram-negative bacteria. Outer membrane is absent in gram-positive bacteria, while outer membrane is present in the gram-negative bacteria. If you talk about lipopolysaccharide, with in short form it's LPS, the content is none in gram-positive bacteria and LPS content is more in gram-negative bacteria. When you talk about lipid and lipoprotein content, it is low in case of gram-positive bacteria, and lipid and lipoprotein content is high in gram-negative bacteria. The flagellar structure, as I said, the bacterial cells, the prokaryotic cell, they show presence of flagella, which is a locomotory organ. They are 2 rings in basal body, while the flagellar structure, there are 4 rings in the basal body. The toxins produced in the gram-positive, they are exotoxins, and the toxins produced in the gram-negative bacteria are the endotoxins. Resistance, physical disruption. The resistance towards the physical disruption is high in gram-positive bacteria, while resistance towards the physical disruption is low in gram-negative bacteria. Cell wall disruption by lysosome is high in gram-positive bacteria. Cell wall disruption by lysosome is low in gram-negative bacteria. Resistance towards drying is high in gram-positive bacteria. Resistance towards drying is low in gram-negative bacteria. Inhibition by basic dyes is high in gram-positive bacteria. Inhibition by basic dyes is low in gram-negative bacteria. Susceptibility to anionic detergent is high in gram-positive bacteria, and susceptibility to anionic detergent is low in gram-negative bacteria. This is all about bacteria, some features of bacteria. In my next slide, we will discuss about archaebacteria and eubacteria, under which you will study the famous cyanobacteria. Thank you so much.